Hello everyone, this is Dr. K. Manohar Babu from Guide School of Pharmacy. In this presentation, I would like to elaborate the role of prokaryotes in pharmacy. First, we will see the different organelles of prokaryotes and then its role and function and applications in pharmacy. Prokaryotes are bacteria is helpful for the production of the antibiotic by providing liposomes for the drug delivery, fermentation process where you can prepare alcohol, various organic acids and specific enzymes to digest substances. So apart from this, it has enormous applications in pharmacy. So this is the schematic representation of the prokaryote or bacteria. The bacteria has various organelles right from the capsule, cell wall, plasmid, etc. So we'll go one by one. First, we'll see the outermost layer is called as capsule. The physical strength which will be provided to bacteria is cell wall. Majorly it is made up of peptidoglycan. The inner layer is called as plasma membrane. It has a thread-like structure called as flagella. Mesosome, which is called as respiratory organ, and it has pili to attach a particular surface. Then it has uncovered DNA or genetic information. Normally we will call it as nucleoid. It has extra chromosomal DNA called as plasmid. Prokaryotes has the ribosomes. Normally it is 70S ribosomes, 50S plus 30S. Apart from this, it has the glycogen as a inclusion bodies, gas vacuole and cytoplasm. All these are the different organelles of the prokaryotes. So we'll see the applications of each and every organelle of the bacteria. First we'll see capsule. Capsule, it is a bacterial capsule is the outermost layer of the bacteria. Normally it is made up of polysaccharide. It is a thick viscous and it will not easily washed off. Normally it is going to give protection for the bacteria to avoid the phagocytosis by our immune cells. The second layer is called as cell wall. It basically made up of peptidoglycan. It's a rigid structure. So this cell wall decides the shape of the bacteria, whether it is a rod shape or cocos shape or spiral. The cell wall is composed of peptidoglycan. So normally the antibiotics such as penicillin are going to work by inhibiting the cell wall of the bacteria. So these are the different shapes of the bacteria: cocci, bacilli and spirilla. Beneath the cell wall, a thin membrane is called as plasma membrane. It is very sensitive. It has a selective permeability character that will helpful for the transport, biosynthesis and energy transduction. So majorly the antibiotics such as polymyxin B is going to inhibit the cell membrane, thereby it is going to show its antibacterial activity. We can see the external structure called as flagella. So this is normally made up of flagellin, a particular protein. It will helpful for the locomotion, for the movement of the bacteria. Here the specific antibiotic such as azithromycin work by inhibiting the flagellar protein in Salmonella typhi. Next we'll say about the mesosome. It is a inner folding of cell membrane, normally to increase the surface area. It is useful for the respiration of the bacteria. It is a site for the respiration and it consisting of a lot of respiratory enzymes. It will helpful for the cell wall formation, DNA replication and distribution of the daughter cells. So normally the antibiotics such as oxalic and vancomycin are act by breaking or inhibiting the mesosomes. So apart from that, we are going to discuss about the pili. This is outermost hairy like structures. So these are basically useful for the attachment of a particular surface. It is composed of a particular protein called as pilin. Pilin is also helpful for the conjugation, for the transfer of genetic material from one bacteria to another. So specific function is to adhere to particular surface. So pili is normally useful for the preparation of the vaccines and monoclonal antibodies against a particular pathogen. In prokaryotes, the genetic material is floating in the cytoplasm. There won't be any specific membrane around the genetic material. Normally it is called as nucleoid. This DNA doesn't have introns in it, and as like eukaryotes, it doesn't have any specific protein called as histones. DNA present as a single circular chromosome, no nuclear membrane around the genetic material. The non-coding sections are called as introns, which are not present in the prokaryotic genome, and it doesn't have any histones. Next, extra chromosomal DNA is called as plasmid. It's a single circular DNA which is helpful for the transfer of the information from one bacteria to another bacteria by the help of pili. Plasmids are responsible for production of various therapeutical ingredients such as human growth hormones, insulin by recombinant DNA technology. Ribosomes of the prokaryotes are completely different from the eukaryotic. So prokaryote we have 50S and 30S ribosome, whereas in eukaryotes we have 60 and 40. So these ribosomes are we can specifically inhibit by a specific antibiotic. Say for example, streptomycin is going to inhibit 30S subunit of the ribosome of prokaryote and inhibit the protein synthesis, while chlorophenicol and clindamycin inhibit 50S subunit of the bacterial ribosome and inhibit the protein synthesis, thereby the bacterial growth can be inhibited. Glycogens are the energy storing molecule present in cytoplasm. From the bacteria, after evolution to eukaryote until the new generation, the energy storing molecule is glycogen from the bacteria to till human. In human, the glycogen is stored in liver. To bacteria. Glycogen is a stored form of the glucose. Glycogen is normally considered as a fast, transient and flexible store of the nutrients due to its highly branched structure. In animal, glucose is going to store as a glycogen in liver. So this is one of the key point that the prokaryotes are the basics of eukaryotes. Gas vacuole is a specific character that is seen in bacteria and even in the plant cells in eukaryotes. So normally this gas will be helpful for the floating the bacteria. Gas vacuoles are generated due to the metabolic activity, and this gas is going to be stored in a particular vesicle where it will be helpful for the floating of aquatic bacteria. So gas may be hydrogen gas or methane gas, normally useful to float in the aqueous medium. Cytoplasm, this is a fluidy substance in which all the organelles are going to float. So it will provide a proper medium for various metabolic activities in the prokaryotes. Cytoplasm is a key component that all the metabolic activities are going to be done. So it will influence cell growth, metabolism and replication of the bacteria. Hope this video is useful. Thank you so much.